one histology slide with a lot of blue dots. Those blue dots are basically the lymphocytes having a very large nucleus, basophilic nucleus. That is why a lot of blue dots, lymphoid tissue. And the options given are maybe it is thymus or tonsil or lymph node or spleen. They all are lymphoid organs. Now, when we are looking at this diagram, they are asking us to identify the structure. Histology slide giving us the options of lymph node, thymus, spleen and tonsil. I'll go with thymus. Now, why would you go with thymus? Thymus is actually lobulated structure and will have a lot of septa which are coming from the capsule, and because of that it will be having lot of lobulated appearance. See, this is the capsule and sending the septa, capsule sending the well defined lobules are there, and these are all septa. And one other thing which you have to find immediately is, if you are talking about lymph node, there will be lymphoid follicles made up of those B lymphocytes. So, lymphoid follicles made up of B lymphocytes should be there in the lymph node, and they are also present in spleen. That is, in the spleen you will have what is called as the white pulp. So, in the white pulp of spleen you will have the lymphoid follicles, and even in the tonsil you will have the lymphoid follicles. The thymus doesn't have lymphoid follicle, and in this diagram there is no lymphoid follicle. So, the very first thing is, if I don't find lymphoid follicle, then it is not going to be lymph node, because lymph node have lymphoid follicles with V lymphocytes, and lymphoid follicles also, if you look at the diagram, you'll find that will be having a germinal center. Germinal center will appear lighter, and the periphery will appear darker. So, these are all B lymphocytes, and B lymphocytes forming the lymphoid follicles. This is seen in lymph node and spleen and tonsil, but it is not seen in thymus. So, absence of lymphoid follicle is basically going in favor of the thymus, plus it is a lobulated structure and lot of septa are seen, and because of that the see multiple cortex and medulla, cortex, medulla, cortex, medulla. So, this lighter area is the medulla, and this darker area is the cortex, and you will see this is medulla, this is the lighter area medulla, in which you can see some of these structures also, which are called as the thymic Hazel's corpuscles. I'll show you the enlarged diagram also, Hazel's corpuscles in the medulla. So, this is medulla, this is the medulla, and outside it is cortex, outside it is darker is cortex, inside is medulla. So, if you see multiple episodes of darker lighter cortex medulla, darker lighter cortex medulla, darker lighter cortex medulla, multiple lobules with that appearance, then it is thymus. And remember, thymus is not having reticular fibers, type 3 collagen. It will have thymic epithelial reticular cells making the reticulum, but there are no reticular fibers or type 3 collagen, and these reticular fibers are definitely present in the lymph node, in the spleen, in the tonsil, but not in the thymus. That comes as a question. So, this is definitely one example of thymus we have to take, and let us look at some of the diagrams also to differentiate the other type of lymphoid organs as well. And you should also know thymus is the primary lymphoid organ along with the bone marrow. Otherwise, rest all, maybe it is the lymph node or tonsil or spleen, they are secondary lymphoid organ. Thymus is primary lymphoid organ because it has stem cells making the lymphocytes. Like in bone marrow you have stem cells you have, then you are primary. Now, these are the lymphocytes we were talking about. A lot of blue dots because they are cells with large nucleus and which is basophilic, taking a lot of blue color. So, this lot of blue dots will be seen in all the lymphoid organs. Now, this is a lymph node. Why would you say it is a lymph node? First of all, lymph node will have a large subcapsular sinus. There is a capsule, and deep to that will be large subcapsular sinus, plus these are the lymphoid follicles. These are the lymphoid follicles, these are the lymphoid follicles, these are the lymphoid follicles, and if you don't see lymphoid follicle, if you see cortex and medulla, if you see this is the cortex and this is the medulla, this is the medulla, then this is the cortex. So, that is basically a thymus. Thymus does not have lymphoid follicles at all, and it is highly lobulated, very fine septa are coming and dividing it into multiple lobules, and this is you see the Hazel corpuscle we were talking about. So, Hazel's corpuscles are basically thymic epithelial reticular cells which are degenerating, maybe secreting some of the cytokines also. So, that is the Hazel corpuscle in the thymus. This is thymus basically. But then what about the lymph node? Lymph node, you will have lymphoid follicles, you have large subcapsular sinus, plus there will be outer cortex, which is basically lymphoid follicles, means B lymphocyte, and then there will be a paracortex. So this is the inner one here. This region is paracortex. In the paracortex, you will have the T lymphocyte, and then there is a medulla. In the lymph node, there is a medulla, and medulla will have the lymph sinuses, because there is a lot of lymph or extracellular fluid which is coming. So how do you identify, first of all, lymphoid organs? By a lot of blue cells, blue cells, blue cells, blue cells, so lymphoid organs. Now, how do you identify one lymph node? Lymph node is having outer cortex, then there is a paracortex, and then there is a medulla. What about the thymus? Thymus is a highly lobulated structure with Hazel's corpuscle, multiple cortex medulla, cortex medulla, cortex medulla, and no lymphoid follicles. Then how do you identify the spleen? Spleen, you will find that there is a capsule sending the trabecula inside. These are the trabecula. So this is the capsule of the spleen, and spleen will have 80% red pulp. So this is all red pulp, and red pulp will have sinusoids, splenic sinusoids, also the cords of Billroth. So cords of Billroth are basically the reticular fibers. As we were telling, reticular fibers are present in all the lymphoid organs except the thymus, because thymus is having these thymic epithelial reticular cells forming the reticulum in required. So reticular fibers will be there in the splenic cords of Billroth surrounding the sinusoids basically. So these are the sinusoids filled with the blood. Along with that, you'll have these white matter. So white matter is very limited, and these are lymphoid follicles with the B lymphocyte. Not only B lymphocytes, but it'll have one central arteriole, and around the central arteriole you have peri-artery lymphatic sheath, which is T lymphocyte. So B lymphocytes, if you are talking about, then they'll be in the lymphoid follicle. See, we are talking about this spares 25% of the spleen will have these lymphatic nodules, and lymphatic nodules also, they are mostly the B lymphocyte, but in those lymphatic nodules, like you can see, this is one central arteriole. This is one of the central arteriole. Around the central arteriole, you'll have peri-artery lymphatic sheath, the T lymphocytes. So T lymphocyte here, B lymphocyte here, this is all WBC's white pulp. This is the white pulp, white pulp, white pulp, white pulp, but white pulp is very less in the spleen. Now, how do you identify the tonsil, like palatine tonsil? Palatine tonsil, palate will have the same lining as the oral cavity. So stratified squamous epithelium. Very first thing you should look for is stratified squamous epithelium. And there are tonsillar crypts. You see these, this is one crypt, this is one crypt, this is one crypt. So tonsillar crypt, and even the tonsillar crypts, they are lined by the stratified squamous epithelium. Plus, you see these are lymphoid follicles, and these lymphoid follicles, as they are marking the lymphoid tissue. So if you want to identify the tonsil, like palatine tonsil, you should have stratified squamous epithelium. That stratified squamous epithelium is not present in other organs. Like thymus, it has a capsule, no stratified squamous epithelium. Lymph node, no stratified squamous epithelium, it has a capsule. Even spleen has a capsule. So presence of stratified squamous epithelium makes it sure that it is, you are talking about tonsil. No lymphoid follicles, you are talking about the thymus. And if you are having cortex, paracortex, and medulla, you are talking about the lymph node. And if you are having 80% of red pulp and 25% of the white pulp with the B lymphocyte and T lymphocyte peri-artery lymphatic sheath, then it is the spleen. Let us go back to the question then. And in this question, we already have identified it is a lymphoid tissue with multiple cortex medulla, cortex medulla, cortex medulla. In the medulla, you can see some of the Hazel's corpuscles also, though not very clear. So of course, it is choice number B. Thymus should remain our answer here.